It's different than last week.
If the interface has changed,
Yeah.
it has, just when you get used to it.
I think we'll just drag it out of the way.
This
We can minimize.
way,
There we go.
yeah, and then hopefully.
Yeah, no, I think it's so active for that. Yeah.
To the hair destination.
And now you should be good to go. They can hear you.
I remember Nancy, on the smooth here 4 months ago.
Little colony. Yeah.
I used to work.
Yeah.
Private market.
Did you change your email address? Everybody now has their Mm-hmm.
Yeah. Standing in the building, watching everything around you collapse.
Like, yeah.
Yeah, it's not. So, I was there 15 years, 10 of those years it was back. In terms of the environment,
Yeah.
there's no better creative environment.
Yeah. Yeah. Yeah.
Without shareholders' responsibilities.
For the longest time though, SRI has worked so hard.
Yeah.
Yeah. I've written this one first, because now you can see
I
all
don't
the
think there's
that.
responsibility. I'm sure I'm going to be asked for some lectures here. But,
Yeah, okay.
nothing can do. I work in the area of infectious
Yeah. That's magic.
I'm in research. So I have no
Yeah, I have trouble right now with Outlook not showing up on my first week. I see guys look at it. They were stumped too, and they said, okay, we'll try our next resort, Googling. I'm like, oh, I did that. That's not what I tried. They're like, oh. I think we need to uninstall Service Pack 1 and there's a hotfix at this link. But I'm not qualified to do that, I think. Uninstalling Service Packs has out of my pay grade.
But,
Yeah. Guess it's our time. So, okay.
I'm a one guy. Story for them. Easy elements.
All right, thanks for coming, everybody. Even in this dreary, gray, fall day. Oh, it's spring. We'll go for it. We have a few more people grabbing their lunch, so they'll join us in a second. But today is a really exciting topic and a really important topic in drug discovery, which is target validation. What makes a target druggable? What makes a target validated? And when you apply for drug discovery funding here at UAB for the Alabama Drug Discovery Alliance, it's one of the things that we really look at very carefully to see if we want to invest dollars in getting your project to go forward. So what the evidence that it's a good place to interfere with in the pathology of a disease. There's one right here if you want to grab it. So we have Mark Sudo present today. He's the Vice President for Drug Discovery at Southern Research. He's been there a few years now. We work together very closely in the Alabama Drug Discovery Alliance, and together we can field a lot of your questions. He's a medicinal chemist by training. I still hope it rubs off on me so that I know if something is a xylene or a benzene. I know benzene, but it's pretty much it. Together we really kind of help shepherd these projects from initial concept to hopefully ultimately getting something usable. I won't go through his whole bio, but he's worked at several different companies, big and small. DuPont Pharmaceuticals comes to mind, Icogen, and he's been here now 2.5, 3 years?
No, since August 2011.
Oh, 3.5 years. So with that, I'll let him take the stage. Thanks.
Thanks, Micah. So if some of you saw this talk a couple years ago, it's going to be very similar. But one of the things that Micah mentioned is probably the most important decision you would make in a drug discovery program or initiating a program is that your target, what that target is and how you go for that target, how you move forward with that target. And it's probably one of the ones that probably, I would say, if you start talking about target validation and when the target is validated, probably has and causes the most controversy. So what I'm going to talk about today, to start off with, is a little bit of what's involved in establishing a discovery project. I'll cover biological relevance, chemical tractability. This is really going to be more on all focused on small molecules. Really looking at when is a target validated and how do you make that determination. I've got a couple of examples that I pulled from the literature and on some programs that I've worked on in the past. The next step, once you find out a target, it really, for a drug discovery program, you'll see the term, is the target druggable? Meaning, is there an opportunity for small molecules to interact with that target? And that comes up perhaps in the aspects that people have not found small molecules for certain types of targets. And I'll discuss a little bit what types of targets people like to work on when they move forward because they're more successful. And then you get the druggable, you get the drug likeness, and that is what your compound looks like to some extent. And I've got a term that I throw in there that you might have a drug that's valid or a target that's validated that is a viable target for drug discovery. So a couple of different examples. Just to put everybody on the same page, we are familiar with hopefully the drug discovery and development. Well, that doesn't work. The pointer, just trying the pointer, doesn't work on that screen. The drug discovery and development pipeline, and you see the first two things that really come to mind are the target identification and the prioritization and validation of that particular target. And as many of you know, this is a long, drawn-out process and costs quite a bit of money to do so. Many times taking almost 15 years to complete the entire process. The numbers nowadays are $800 million to $1.3 billion in a drug discovery in a large pharmaceutical company. To be quite honest, if you were doing it all by yourself, it doesn't take that much, but they actually put in all of the various failures are included in those particular numbers. There's a lot more. Why is it so expensive? There's a lot more regulations and requirements. There's a large failure rate. And really, it's still to the point where 1 in 10 compounds entering phase 1 make it to a drug. So you're getting to a point into phase 1 where you're putting compounds in, 90% are failing, and it's costing you an awful lot of money. So some averages that people have used in the past, if you start 100 brand new screening programs coming forward, new targets, 50 of them might move forward and you'll be able to find some leads. What I mean by leads, some chemistries to do some chemistry on that are tractable. The 20 to 25 will advance into late-stage lead optimization, and probably only 10 programs or so will move forward. So your success rates are relatively poor. There's been a lot of ways that people have thought to try and improve these success rates, put parameters around these programs earlier on. There's been more and more parameters putting into how some of these programs are accepted and why. So why do compounds fail? Why is the failure rate so high? And I call this a changing paradigm. I think several years ago, if you looked at some of these numbers, pharmacokinetics was a very high reason for failure rate. There was not a lot of studies done preclinically. We didn't understand perhaps some of the models that are available. We didn't have some of the in vitro methodologies to be able to predict better of what's going to move forward. There's a lot of work done now trying to correlate when you're looking at pharmacokinetics and some of these parameters like absorption and distribution, metabolism, excretion. Where is the compound going? How long is it there? What compartment is it in? If I take a look at doing, for example, an in vivo pharmacokinetic study, how does the half-life in a rat going to correlate to what I might see in a non-human primate or a dog? How might that correlate to a human? So that was the issues. And I think right now we've done a much better job. So there's probably less compounds starting to fail. Or less compounds failing now due to pharmacokinetics. There's also other opportunities where you can do phase 0 type of pharmacokinetic studies with very small amounts or microdose studies in humans directly to understand before you move forward. Toxicology, That still is an issue. It's very difficult to predict some of the toxicities you see in people from what you see in animals. You always run into the effect that you are looking at rodents with a lot of adverse effects in people that are not predictable. Still a big one is lack of efficacy. Biological rationale is incorrect. Animal models, for example, arthritis model in rats versus rheumatoid arthritis in patients is probably one of the better types of models, because many of the compounds that work in, for example, the arthritis model do work in people. And the oncology models of those who work here have not been very successful in predicting what's really moving forward. Sometimes, I throw this in there, sometimes a project fails for commercial reasons. An organization decides it's not commercially feasible, there might be competition, they've moved out of a therapeutic area. So, this is something that's been published recently, and I have a copy of this, and it's a very, very good article if you take a look at this article. AstraZeneca went through their pipeline very recently, in the early, would have been probably 2008, 2012 timeframe. And a matter of fact, Bob Bostwick, who's our head of high-throughput screening, was at AstraZeneca at the time they were actually doing this analysis. They really did a strong critique of why their programs failed. And they show you the reasons, not only from efficacy, commercial reasons, reasons that might be considered personal reasons. I need to keep this moving forward because my bonus depends on having this program. But they have in here what they call the 5 R's. You have to have the right target, which is the first thing right away, the right patient, the right tissue, the right safety, and the right commercial potential. They also threw in here, which is very interesting, the right culture, how you actually handle these things within an organization and move forward. If you really want to read an interesting article, take a look at this article. Another article that just was published recently, validating molecular targets, and I wanted to point out here, I don't have a pointer, but somewhere in here, if I can find it, half the drugs fail due to insufficient efficacy. In other words, the clinical target validation fails for about 50% of therapeutic approaches. So it really is still a big issue. Oh, I had it on the bottom. The clinical target validation for about 50% of the therapeutic approaches. And this is European Drug Target Review 2014. Will that one work?
How many hope?
No, see, it's the,
Oh,
this is where it puts the screen. If the screen absorbs, see, it works over here. I could point here, but. All right, just to go through, for those of you who might not be familiar, preclinical drug discovery starts off with target identification and validation. Typically, in a lot of the programs, we start off with a high-throughput screen of lead identification. This is looking at in vitro activity, your IC50, your EC50. It goes through, do we have chemical tractability? Are there liability targets like the CNS, cardiovascular? There's a med-chem approach and lead optimization. Right now, we throw in this a lot of in vitro ADME prior to doing anything. In vitro means we're doing liver microsomes, solubility, permeability studies. And if you're involved in one of the ABA projects, we're routinely generating that data for those projects. Now helps us to actually guide structure activity relations and medicinal chemistry properties before we start doing, even doing any type of in vivo assessment. Toxicology and additional liabilities, in vivo assessment, as I said. In many programs, you get to a point where you have very good active leads from the chemistry perspective. Your biology seems to work. Your potency is where you need to be. You still need what I call that last step is to in vivo optimization to really try and work on parameters to either increase bioavailability, tissue distribution, or get the various compounds to the site of action, for example, the CNS. So the key steps is the target validated and druggable for small molecules. How will you screen for modulators? Are you looking for an agonist, an antagonist, an inhibitor? The outcome in general you're looking for. Many people forget, well, where is my target? Is it in the CNS? Are the liability targets in the CNS? So the compound could be in CNS or not, is in the periphery. Another important aspect is the secondary assays and species differences. You may be running mouse or rat. What's the correlation to human? Can you set up those kind of studies as well? I've seen example in ion channels where compounds have been very, very potent against rat and not hit the human channel. And I've actually seen the opposite, where compounds were very, very potent against the human channel and not hit the rat. The animal models. Are there standards or known compounds? The predictability of a particular model. Have compounds that have been approved worked in these models? A very important aspect now is a biomarker. Modulating your target will that lead to some kind of indicator that can be measured readily in a phase 1 clinical trial. So you're not doing efficacy, but you're looking for some type of indication that you're getting target engagement in the clinical population. And then the clinical outcome. So failure rate, right? Alzheimer's disease. This, for example, has a higher failure rate, clinical failure rate than any other probably therapeutic area. There's not a single disease-modifying drug approved. There's lack of clear animal model that recreates the pathological and neurodegeneration hallmarks of AD. You may have to target particular areas of the brain. So all of the various targets that we look at goes to the question, are the targets not validated, or is it really due to a lack of an animal model? I think most people, maybe who you talk to, would be either or. Probably would believe right now that the targets are probably valid. They seem to have some association with the disease process, but the animal models that we're using are not relevant to be able to pick up what we're really looking at. When I talk about a target, I found out I was having a conversation yesterday. What is a target, right? It's a protein, a receptor, an enzyme. It's either inside the cell or on the cell surface. For example, a G protein-coupled receptor, protease, or a kinase. Some of your targets are transcription factors. You're looking at gene regulation, ion channels, transporters. When you look at infectious diseases and cancer, many times they were almost directed cell-based assays, right? Phenotypic assays looking for cell kill or something else happening, and then you go in and elucidate the target. And then you have what are probably considered some of the most difficult druggable targets. Could be valid targets, but druggable targets are protein-protein interactions. So here's a case history. This compound, Senicapoc. It's a potassium channel inhibitor for the treatment of sickle cell anemia. It blocks a potassium channel, IK1 or IKCA 3.1, and with this inhibitor in red blood cells, by blocking this channel, maintains the red blood cell and helps with preventing the dehydration that occurs. So this advanced to a phase 3 clinical trial in sickle cell patients. We had a positive phase 2. The phase 3 now, though, we had a positive phase 2, and that was based on hematological factors. When it went into phase 3, the FDA said, well, your approvable endpoint is no longer the hematological factors. You have hydroxyurea, which is your current therapy. You need, with your drug, to actually show that you're going to decrease the amount of vaso-occlusive crisis rate as an approvable endpoint, which is very difficult. All patients going into this particular study had to have a certain amount of occlusive of these crises occurring a month, even though they were probably on some kind of standard therapy. So there was 3 arms to the study: our compound alone, hydroxyurea alone, and in combination with hydroxyurea. An independent review board analyzed the data. There would be no benefit. The trial was stopped. As a small biotech company, our stock went down. There was an improvement in several hematological factors, right? So the question really becomes, is this a validated druggable target? It might be. Do we have the wrong compound? Did we do, could you have gone forward and perhaps looked at a different endpoint? But those were the parameters placed upon us. So at the time, we probably thought it was a pretty good target. So where do these ideas come from for these targets to start some of these programs, right? Natural products, we've got a lot of natural products, medicine, old wives' tales, herbal medicines. We get ideas for targets and moving forward because there's observed effects in animals. I started my career several, I guess it would be decades ago. There was not a lot of molecular biology. Sometimes a chemist would make a compound, find a biologist who had an in vivo assay that was ready to go, give it to him, and all of a sudden I'm working in hypertensive agents because of decreased blood pressure. Now we're into more rational approaches, right? Biochemistry, biology, screening systems biology, understanding genetic mutations in people. I mean, honestly, I've worked on targets in the past that we knew very little about particular targets and what was happening. We've got a better understanding, at least we try to nowadays, about what our targets are doing. So, there are new targets, right? That we've identified more targets probably than we know what to do with. It's probably the problem. But it's genomics, proteomics, pharmacogenomics, RNA interference, knock things out, knock things in. We got pathway analysis, we got transgenic animals, but the key question, right, is still the target validation. Modulate that target, and what effect does it have with a small molecule? So a lot of these are good, but I'm not quite sure that they really get to the point of saying, just because I can knock down the target with siRNA and I see some type of effect, it's actually a valid target for drug discovery, or is it a drug target for drug discovery as well? So what really does constitute a validated target? Well, you can have a genetic mutation, right, leading to or associated with disease. For example, Alzheimer's amyloid precursor protein secretase seems to be a genetic mutation, but yet we've had no success in finding drugs for that. Human epidermal growth factor, amplified in breast cancer, promotes cancer cell growth. That is probably a validated target. Upregulation during the disease process. How about, I like inflammation in cyclooxygenase 2. Vioxx, all the COX-2 inhibitors look like a really great way to move forward, for example, right? It revolutionized, people were drugs, out there from Merck, from Pfizer, that were COX-2 inhibitors. It was a really big market at one point. And then another thing, validated target, people like to look at the proteome. They like kinases in general, and I kind of throw in here, I'll talk a little more about gene family. So you see a lot of drug discovery projects in kinases, right? People are comfortable with kinases. They feel very good about kinases. They talk about a validated target, maybe. KCNQ23, this is an ion channel, a potassium channel, and it's present in high levels in neurons, including dorsal root ganglion. There's no significant expression in major peripheral cortex. It's associated with benign familial neonatal convulsions, where children get this, they grow out of it, but people that are children do have this have a higher propensity to get epilepsy of some sort as they get older. Targeted deletion of KCNQ in mice increases sensitivity to chemopulse agents, and there are small molecule activators that have efficacious in animal models. So this might be a valid target. So you look at it overall, what you have to deal with. You got a KCNQ family. So it's a valid target, but there's 5 members of this family. And what you got KCNQ1 is in the heart, so it needs to be avoided. 2 and 3, as you can see, form heterodimers and work together. There's KCNQ4, which is mostly in the inner ear, and nobody worries about that too much because it's difficult to get drugs in the inner ear, and KCNQ5. This is a typical, when you're looking at this type of target for an epilepsy drug, where a typical progression pathway, you do your initial screening in vitro, SHSY5Y cells, for example. In this particular program, we counter-screen for KCNQ1 because right away we do not want to deal with cardiovascular issues, L-type channels, calcium, hERG, etc. And this is kind of a typical pathway analysis that we would do, that also we would do in the ADDA, and you make some decisions moving forward here for this. So this is an actual progression pathway. We found compounds that actually work in this particular area. We moved them into clinical development with a compound that actually was approved called retigabine that works in this area as well. But there's another interesting story, if we have time, that I can discuss about this particular channel and some of the untoward toxicities that we saw in the liver associated with this particular channel in 4 or 5 different series of compounds, even though we did not think there were very high levels of this particular ion channel. Cyclooxygenase inhibitors. So is this a validated target? So if you remember, the classical COX inhibitors, right, they're not selective. They inhibit COX-1 and COX-2. And what happens is the resulting inhibition of prostaglandins and thromboxane synthesis, they reduced inflammation, and they were antipyretic, antifibrotic, and they had analgesic effects. But they really caused big problems in the gut with ulcers, and people bleed, and some people, if you take a look at some of these now, really have bad reaction, have died. And some of them are also acidic, if you knew the structures, and cause additional damage to the GI tract. So in the 90s, it was discovered there was COX-1 and COX-2. COX-1 is known to be present in most tissues, as you can see here, maintains a normal lining of the stomach. So the thought was, well, blocking COX-1 is not a great idea. I've got COX-2, which is primarily upregulated inflammation. Therefore, all I need to do is find a selective COX-2 inhibitor, and I got a great drug for inflammation. They're both associated with some of the same pathways, resulting in pain and inflammation. And really, the idea was that COX-2 is considered desirable, COX-1 is not. A COX-2 inhibitor, celecoxib, rofecoxib, as I said, a very large market. If you look at this story, what was happening, probably in the late 90s, there was a rush between Pfizer and Merck, and who was going to get the first one out. I think Merck won with Vioxx. But they were still, at this point when they were being sold, $2.5 to $3 billion. What was happening is it was brand new. People thought it wasn't going to have the side effects hitting this particular target than the others. It was probably overprescribed a bit. There was an analysis done, and to make a long story short, they found out that there was increased risk of vascular events, mainly due to twofold risk of myocardial infarction with the COX-2 inhibitors. So, as if you know the story, the drugs were withdrawn at some particular point. Some of them are still available, and part of it was they were being overprescribed. So, is this a validated target? Is it a druggable target? Yes, you'd say it's probably druggable, but is it validated? And how do you really determine it's validated?
Can you make money off of it?
This may get into a target that it may just not be a viable target.
Well,
It might be validated, right? It may be druggable, but it might not be viable from what you're going for because of the other effects that are associated with it. So it's an interesting kind of dilemma. And part of this is just to raise some questions when you think about some of these targets as to what's happening. So sodium channel inhibitors, right? There's a lot of sodium channels out there. They're probably considered validated targets. You have your local anesthetics, lidocaine, procaine. They're worked for epilepsy, you got phenytoin, you got antiarrhythmics. They're used for neuropathic pain. All of them are non-selective. All of them affect multiple channels. Are they validated targets? These compounds, if you look at them, are hitting a lot of sodium channels. NAV 1.7, 1.8, probably 1.3. Some of the effects you're seeing because they're hitting NAV 1.5, which is in the cardiovascular, NAV 1.2, which is in the CNS. So you really have very non-selective compounds. And you take a look at the structure, remember that that's maxillary. However, if you look at, here's all the various sodium channels that I was actually showing here. So you can see where they are and how they're used. And there's anti-epileptic agents that block these channels, local anesthetics, etc. If you look at NAV 1.7 and 1.8, two sodium channels with some recent data. So there's conditions where you have congenital insensitivity to pain. So, and they have SCNA missense and in-frame deletion mutations. So NAV 1.7 or SCN9A several years ago really was a very, very hot target for neuropathic pain and a number of indications. So there's mutations in this gene that cause 3 human pain disorders. Biallelic loss of function mutations and channelopathy associated with insensitivity to pain. So therefore, people with certain mutations do not feel pain. There's certain families that do not feel pain with these. I mean, you might see people walk across the fire. I mean, they really have identified that they do not feel pain. There's a couple of other disorders where people are extremely sensitive and have certain areas and much more pain all the time. It's paroxysmal extreme pain disorder and primary fibromyalgia, right? So it's mutations in the other direction, so they're always getting pain. So you've got 2 different mutations showing one that decreases pain, one that increases pain. So this seems like, a really, really great target. What I like about this one, so what happens to someone if, and it was a concern when we were working in this area, if I have a NAV 1.7 or an SCN9A blocker? One of our concerns was the person has an arthritic joint. they're taking one of these compounds, they walk over, put their hand on the stove. Would they feel pain? It was somewhat of a concern what the effect would be with a small molecule modulator of these targets. All right, so here's the question. When is a target validated? When do you start? Just with in vitro data? So overexpression, NXN mutations. Do you need cell-based activity to say it's a validated target? Animal studies, knockout studies, disease phenotype, therapeutic interventions. So do I believe it's a validated target when I have a small molecule that blocks it? Or a phase 2 clinical result? Or an NDA approval? I think you'll probably talk to various people and you'll get a difference of opinion. So when really, I mean, it's still a good question. When is a target really considered validated? My opinion? Depends on who I'm talking to. So, you know, I feel pretty comfortable about, with the drug discovery program, in my mind, if I think we can get a small molecule that's going to work in a relevant animal model. Right? It becomes the importance of the animal model. I think then, is that animal model, I'd like a little bit more proof that that animal model has a lot of relevance in the clinical situation. If you work in infectious disease or antivirals, that's a little bit easier. If you work in neuropathic pain, maybe not so easy. Some of the diseases associated in neuroscience areas, the behavioral models associated with it is very, very difficult. If you have something you can measure readily with a biomarker or antihypertensive or glucose levels, you feel a little bit better. So it really depends. But I would probably feel comfortable with myself making sure that if I got what I would consider maybe a chemical probe or a small molecule that modulated the target in a relevant animal model, I would be comfortable with moving forward. But that's my opinion. And I would look for really some type of, early on, if we had a biomarker that showed that I was involved in it. Now, just to a little bit to carry on further, I also want to show that in this animal model with numerous compounds, that the potency of the compounds are related to an increase in potency in vivo. There I could show that that compound is interacting with that particular target in some way. So I could generate a pharmacodynamic type relationship. So that I really see that the compound itself is causing the activity in vivo. So here it is, validated target versus druggable target. This is the next big step. So is there a difference? The answer is probably yes. A target could be druggable, but it may not be validated. A target could be validated, but it may not be druggable. And what is a druggable target? So most people would hope that a druggable target is a validated target. So you have different types of drugs, right? Natural products, steroids, peptides, biologicals, small molecules. Oral bioavailability is really probably preferred, ease of manufacture, stability, and cost. There's a lot of work being done in biologicals these days as well, especially in the cancer area. Okay. A new validated drug target, issues to consider. So I've got my validated target. Is the target druggable? What evidence is there to support that initiating a drug discovery program? Why should I do this? I've got the target. What other evidence is there? Small molecule inhibitors, animal models, species differences. You have to really be careful with species differences and a biomarker. I think what's important, what really a lot of people look at, is my target related to other targets. People have been able to work within the gene family, for example, like I showed ion channels. And does that work? You feel a lot better about doing that. And you'll see then like kinases, phosphatases, and nuclear receptors. Also, do I have some structural information, protein crystallographic data, NMR structure, to help me with this particular target? I'm going to have to do a big chemistry program. I'm going to commit a lot of resources forward. How can I use this to guide things forward? The other thing you might consider if you were working in a small biotech or a company, at one point, is there intellectual property around this target? Can I compete in this area? Or if I really want to do drug discovery, is everyone way ahead of me or do I have kind of a novel approach? So, gene families. I wanted to bring this up because when you talk about validated targets, there are certain gene families that people would consider validated targets. Right? A gene family is a group of genes that share important characteristics. or similar DNA sequence building blocks. And why this is important is that there's groups that were set up as gene family drug discovery groups, and programs and expertise directed toward a particular set of targets. For example, years ago, GSK in RTP used to have a nuclear receptor group. They were probably not associated with any therapeutic area. Someone would bring in a nuclear receptor that they thought fit into their therapeutic area or was important. They would find modulators, agonists, antagonists, whatever it might be, for those nuclear receptors, do the studies and the chemistry. They were very good at looking at it. They had specialized libraries that they used against the gene families of interest. They would then provide those molecules to the therapeutic area to further triage the particular target of interest. Whereas you've got, and why that's important if you're doing something like nuclear receptors, G protein coupled receptors, or ion channels, you have all the assays in place to do the counter screens. You don't have to set things up. It's right there. You've got a lot of your compounds may have already been screened. You've got a database, and we've been successful. And here I just list a few of the types of gene families and druggable targets. I've worked at G protein coupled receptors, kinases, worked in a lot of those, mostly all of them. So let me talk about another interesting example in the ion channel gene family. So when you look at ion channels, I'm going to focus on, there's a number of ion channels I have shown here. I'm going to focus on the calcium channels at this particular point. And you can see the types of channels that are there. They're interesting targets for epilepsy and pain based upon function, distribution, and pharmacology. If you look at G protein coupled receptors, so I showed you the ion channel, people work on them, G protein coupled receptors. You look at the amount of dollars that people feel are relevant, they're going to earn from working in G protein coupled receptors. It's tremendous. It's a big, big area. There's still a lot of drugs at the G protein coupled receptors. You know, if you look at G protein, the drugs that work in this area, you got the Zantac for ulcers, you got beta blockers for hypertension, you got asthma, serotonin agonists, you know, for migraine. Remember the ion channels? So you look at what's happening here, you have some of these structures. What you really have is commonalities between many of the G protein coupled receptors and the ion channels. So even though you're in a class of drugs that look interesting, and that's due to the basic amine that you block. So you need to then counter screen many times some of your G protein coupled receptor drugs for other classes of compounds with the ion channels. One other problem, hERG blockers. Everyone's heard of hERG, right? It's caused issues with cardiovascular, disorders, the Plant syndrome, long QT, where people's drugs have been taken off the market due to cardiovascular effects. They also have basic amines. So even though you've got validated target G protein receptors, that particular target to move into means you have to set up all the various assays for GPCRs, you probably have to set up assays for ion channels, and you probably have to be very concerned about some of the cardiovascular ion channels that you're dealing with as well. So a validated target, a very difficult area to work into, and probably takes a lot more work, perhaps, than if you might work into a nuclear receptor. Kinases are the same thing, right? 500 and some odd kinases, whatever the number is these days. You've got to do a lot of screening to work in some of these areas. So let me look at a calcium channel example. I've worked in ion channels. So if you look at T-type channels, there's three family members in T-type: 3.1, 3.2, and 3.3. And ion channels, they represent, you know, some great targets. L-type, you got amphetamine, verapamil for cardiovascular. N-type, you got sconotide for cancer pain. And then T-type, zonisamide, and ethosuximide, and mifeprevil have been used for epilepsy and pain. So calcium channels are considered validated targets and druggable. We wanted to look at T-type calcium channel antagonists. And in the literature, it suggests that T-type calcium channels are involved in certain pain and CNS disorders. There were some small molecules that were used, there was some antisense, there was rodent knockout studies. So we wanted to start a program. So we wanted to move forward and identify a novel series of T-type calcium channel antagonists and evaluate them in rodent models of pain. We were more concerned with pain than some of the other areas I was working at. And we thought it would have a great opportunity because no one was really looking at those particular channels. We had set some criteria. We needed a target. We were working on, this is what our thought was, target validation, right? There was indication the literature was validated. How were we going to validate it? We were looking for an IC50 against our channel versus the plasma brain concentrations. So the idea would be what was critical. How much of that compound could we get into the brain? How much of that compound is free drug? What's the IC50 in order to feel that we validated the target? It had to have, we thought, less than 50 nanomolar, 30, 40-fold selective over L-type to start. We needed oral bioavailability. And what I was talking about was that 5 to 10-fold over the IC50 at Cmax for 1 to 2 hours. That was our target concentration and what we needed and what we thought would be a chemical probe, for lack of a better term. There was a lot more in the literature about these. They worked in pain, epilepsy, arousal states, and even in oncology. Although I could never get any of the oncologists over at Southern Research interested in looking at T-type calcium channel. I don't know why. So, I mean, it was published. I mean, a couple of these publications were by Merck, where they could actually work for a very rare form of epilepsy in this particular case. So we did identify small potent molecules as T-type agonists. They were pan-agonists, and they had 3-1, 3-2, and 3-3, but we didn't think that was an issue. They were very selective versus all of the other ion channels, the cardiac channels. They had very good in vitro properties could be achieved. So, for example, we had permeability, stability, solubility. We had oral bioavailability. We looked at CAPO2. We had limited CNS exposure. However, the plasma concentration versus T-type IC50 were insufficient for pain indications. So what we did is we had a compound that we got moving forward. We achieved what we were looking for with regards to potency, and we got to a compound that was 10-fold over the IC50 in the brain, and we tested this compound in 5 pain models. It was not active. We stopped the program. We feel we did not validate. We set criteria to validate the target. We did not meet that criteria with small molecules, and we actually stopped the program. So, I mean, there could be other ideas to move forward, Parkinson's, whatever. The target was druggable. You find a small molecule. Was it validated? We said no. And just to conclude a little bit, I think it's important to talk about drug-like molecules when you're talking about druggable and validated targets. You see a lot of times various parameters. There's a lot in the literature. There's a rule of 5. It's probably 4.5. Molecular weight less than 500. So a small molecule. Molecular weight less than 500. C log P less than 5. Maybe no more than 1 violation type of idea. You're looking at polar surface area, number of donors, etc. So a lot of parameters in place around what we consider a very good small molecule or drug-like. Drug-likeness, what does a compound look like? What does a drug look like? So you have to have solubility in both water and fat. Orally administered, has to go through the intestinal lining, in the aqueous blood, penetrate the lipids, cellular membranes, and we use C log P to estimate that. What's very important, you'll see differences in drugs when you talk about druggability and drug-likeness is the potency of the drug and what you can get away with. So I've given you parameters about what druggable or drug-like properties are. A lot of that depends on the potency. If you're administering a compound at a daily dose once a day of 5 mg per kg versus a compound, a drug that you're going to administer, say, BID at 500 mg per kg, you can accept a few other little issues with your 5 mg per kg per day compound than you can with your 500 mg per kg. The more compound, the more liabilities, the more toxicities, the more chance for just idiosyncratic reactions to these types of compounds. So you want a compound that's very, very potent, and if it is, it reduces the non-specific off-target pharmacology, low clearance, high potency allows for low total dose. The less you give, the better. So a lot of times you'd be thinking, why can't I be up to 500 or even sometimes I'll see some colleagues, I got something really excited at 30 micromolar. Think about if you're at 10 to 30 micromolar in a cell, the concentration you need to achieve in vivo to be able to get to those concentrations. If that's the best you could get, I would say that's probably going to be very difficult target to validate, and you may have other issues associated with the activity that you're seeing, or there may be other reasons why the compound is looking exactly the models that you're looking at. You can go some really kind of cool ideas about drug-like space and where you fit in design compounds and how it moves forward. There's oral drug space, there's protein space. Even the people doing gene families have designed, kinase space. For example, we've actually used in the LAR2 program earlier on, we used a kinase library. And what that means is they took kinase inhibitors and came up with structures that were somewhat that they synthesized, but computationally looked like kinase inhibitors. And then there's ways to actually guide lead optimization to take into account all of these drug-like properties. You can look at ligand efficiency, philic efficiency. What you really need to do, and I like to show this everywhere, is avoid substructures that know their chemical pharmacological properties. When you do screenings and do assays, you will find, many of you here have done that, you will find compounds that are active that contain nitrogen. I guarantee it, because they're very active in most assays. They interfere with a lot of free radicals. They just seem to be very, very good. When we do some of the programs now with the Alzheimer Drug Discovery Alliance, and you run a screen, the screen is run, counter screen is run, that usually then takes into account running getting an IC50 or an EC50. We run a counter screen against the cytotoxicity or liability target. We take all of those compounds, do a cutoff based upon, say, potency or efficacy, what we're looking at. Then we do a chemistry triage. We eliminate compounds that we know might be promiscuous. We eliminate structures that might be active in a lot of different assays. I mean, I've been in some of the programs that I wouldn't work in that one because it's active in a lot. Now, when you buy a library, it's hard to extract out all of those single compounds because many of the companies sell you libraries of sets of compounds. And we've got about 80 different compounds for southern research that we have to throw at something. But, so you really need to avoid, enones or alkylating agents that really cause a lot of problems. So they would not be considered, very good leads to move forward. Let me just say one other thing now. Natural products. Very effective as drugs. They've been optimized already by nature, right? They don't fit this whole concept that I just talked about right now. and because they're very complex, many of them have multiple chiral centers. You probably couldn't synthesize them very easily. They're difficult to work, but they've been actually making a comeback lately. There's been, and why is in the past people would take natural products, they would go into, say, South America, pick up various parts of the plant, do the extracts, and screen them. You could screen them blind, then you try and do a separation, you have an activity. So it's a very difficult process. And now people have been going forward, and they learn a little more, so they're doing in some ways, for example, a pre-purification. Probably find ways to eliminate a lot of the various byproducts, for lack of a better term, that they find in natural products. For example, Bill Fanicol out at Scripps has gotten some natural products and very, very deep in the ocean floor. And they're natural products, but he's actually worked on, there's probably only 4 to 6 components versus 35 or 40 or 100 that you find in other natural products. So they are making a comeback. People have found we have better synthetic capabilities now. You have a complex product. We have some of them that start from natural products and then we expand on. So there is kind of a little bit of a comeback for natural products, but they don't really fit. Streptomycin does not fit kind of the small molecule, no chiral centers, no reactive moieties type of structure that you might find with a molecular weight of 581.
If that would have shown up as a hit, it would have been like,
Yeah, that would have been gone. Yeah. Hopefully that's probably not in our library. But anyways, so they are, and I just wanted to throw that in there. There are other things that are coming back, and molecular weight at 450 with no complex centers is not necessarily the only way to go in some of these areas. With that, I will stop, and I'd be happy to answer any questions. Thank you.
On the automobile target base, 14 action.
Right.
So can you comment on drugability of 14 actions?
So I think years ago people would say, oh, that's a bad target. Okay? We've got a target in 88 tau 10 with Eric Roberts' protein-protein interaction. So we found hits. If you think about it, it simply is, I should say, it really is a binding pocket of some sort, right? It may be a little bit more complex, but it is a binding pocket of some sort. And what people have done now is expand a little bit and look at some different molecules. We have a collaboration with a company, and it's a long story, so I'll just kind of go through it, where they had come up with, I would say it was a new set of libraries that presented like small peptides, behaved like small molecules. They had a compound from this type of libraries that were in clinical development. It was kind of a peptide-like with a scaffold. Molecular weight was less than 500. The compound was 80% bioavailable. It had all the properties of a small molecule. We had actually used those and screened them against a couple of targets, and they actually worked very well, especially in some protein interactions. So I think now people have looked at protein-protein interactions. Another way that's developed that was not possible, I think what was complicated protein-protein interactions, we wasn't quite sure how everything fit together, right? And you could say, well, I think it's here and here, but there's probably different parts and different parts of the protein come in. So people have been using various fragment-based approaches, both fragment-based approaches from NMR and fragment-based approaches from X-ray crystallography, where you take very small molecules, probably with molecular weight around less than 200, maybe 220, and you soak them into the crystal, or you look at an NMR if you set up the techniques, and you're looking for very, very weak interactions. Only trying to maybe measure interactions with the NMR. It's in there someplace. And then you screen, and you perhaps find different molecules that have different types of interactions. Then you combine those based upon, say, the model that you have connected to design a brand new compound. So there's been a lot of work done through Vanderbilt, for example. I know a couple people that have done work looking at protein-protein interactions and using some of these fragment-based screening approaches to kind of perturb out small points of interaction and go forward. Wei Zhang at Southern Search has been doing, he's not going to do a protein-protein, but he's been doing virtual screening, trying to identify potential binding sites. And he could screen then 11 million compounds virtually for a number of years, look for how to store them, and then try and buy some compounds. And he's done that on a couple of other allosteric sites. And we work that with it's actually applied to protein-protein interaction. So it's becoming, I think, not easier to do, but probably doable if you really want to learn. And we recently brought in-house through another program. We do have a fragment-based library that we have brought in that we have screen. You could do a regular screening, but you'd have to really look for inhibitors of 100 micromolar. I mean, sometimes you're very, very high, right? Because you're just looking for very weak inhibitors that you can come up with.
How does your thought process change when you're thinking about intracellular targets versus extracellular targets?
I don't know. I mean, I still worry about, I can get into the target. I figure I can in some way, perturb that target. I'm more concerned about valuing the target than the target. I mean, there are compounds that get into cellular targets and work. So, I don't consider that an important aspect when I look at it. I worry about, if you have somewhere on the cell surface and what the parameters can be for getting. It might have to be different molecules and what you have to do to make sure you're getting into the cells and hitting the target.
The virtual screening you have mentioned, is it the particular therapeutic category based, or you have identified some targets or very specific?
The screening that we have in Southern Research?
Yes.
Is it the virtual screening or any screening?
The virtual screening.
We can do that in any target. We've done it for oncology targets, we've done it on kinesins, we've done it on viral targets more recently as well.
But you need the structure.
You need the structure. Yeah, you need
structure as of both.
Sometimes you can actually use a homology model if you really want to wing it. So you might not have the structure of the protein you were looking at, but you might be able to use a homology model if you feel you can actually generate some kind of structure that might work. I mean, that's a little bit iffy, but we actually did a collaboration with Ralph Sanders on heparinase, where he had a collaborator that had crystal structure of heparinase, and we did a virtual screen and found leads, small molecule leads, by doing the virtual screen. We only did 5.5 million.
How long does it take to do?
It takes a while.
Just press go and then you go eat bonbons?
Yeah, you go somewhere else.
But if it is a million one, but if it is a 10 to 15, then it should not take that long.
No, it doesn't, no. I mean, you set it, I mean, really, it's not that much, you know, you set it up, you let it run, you go through the results. Yeah, it's not so labor intensive.
So does every product start with in silico?
No, not at all,
screening?
no.
So how, I mean, if you talk about millions of compounds, how do you,
Well, we don't do it.
I mean, how do you start?
Well, we
Let's say I have a hot protein, I
Yeah.
think it's important.
So the way we would start it is we take a look and find out if we could set up, in this particular case, an assay. And what does that assay look like? And what might be the throughput of that assay? What might be the readout? So we actually sort of the assay and say,